Welcome to the Nimble American Rescue Plan webinar series. This webinar series highlights the projects funded through the American Rescue Plan. The goal of these projects is to help the nation prevent, prepare for, and respond to coronavirus public health threats. In this webinar, we will hear from Stephen Stretfield on the project development of scalable, thermostable, spray-dried vaccine formulations applicable for coronavirus vaccines, which would allow for quick and less expensive vaccine distribution in the event of a future coronavirus outbreak. You can find additional webinars in the series on our website, www.nimble.org. And now we will turn it over to Stephen.
Thank you, Daniel. So this project funded through Nimble to Fraunhofer USA, looking at scalable, thermostable, spray-dried vaccine formulations. So most licensed vaccines are liquid formulations. These typically require storage and transport in a refrigerated or frozen state. And therefore, they can be problematic and expensive for widespread distribution, particularly in many developing nations. And it does also result in a significant amount of inventory often being discarded. So lyophilization is currently the main approach to generate dry formulations of vaccines or other drugs that are inherently more stable than liquid formulations. However, this is a very lengthy and energy-intensive process, therefore quite expensive. And it also subjects the vaccines to multiple stresses that can lead to protein aggregation and loss of potency. Spray drying is one of the leading alternative approaches to generate dry formulations. This is a fairly rapid process, a single step allowing for reduced processing time and energy consumption. It's also a controllable process that allows for improved dry particle attributes. And it's scalable and can be joined in line with other processes, so is amenable to drug manufacturing. And we previously demonstrated under other programs the potential for spray drying for protein subunit vaccines targeting anthrax and influenza virus. So just in summary of those introductory points, desire to go from liquid formulations to dry formulations that are inherently more stable. And then in terms of generating those dry formulations, to go from a generally more expensive lyophilization process to an alternative process. In this case, we're looking at spray drying. So under this project, we're going to look at demonstrating the applicability of spray drying to two particular vaccine candidates which were found of relevance. One is a protein subunit vaccine candidate that directly looks at the receptor binding domain of the spike protein of SARS-CoV-2, the causative agent of COVID-19. And then also, we've done some development on virus-like particles and to look at a sort of somewhat more complicated architecture in a VLP, where in this case, a antigen of the parasite, Plasmodium falciparum, causative agent of malaria, and a key antigen there is displayed on the surface of a virus-like particle. The major tasks under this project were to generate this receptor binding domain and virus-like particle vaccine antigens and spray dry them and characterize them. We did this using a range of solid state assays to look at the solid state and then assays to look at the reconstituted vaccines in the solution state. So looking at both dry particle characteristics and also biochemical attributes and bioactivity of the vaccine candidates. So as a first round, we generated 24 spray-dried formulations for each of these vaccine candidates with a range of different excipients and spray drying conditions, and with and without adjuvant present, and then performed stability studies on these various formulations at a range of temperatures up to and including 60 degrees Celsius. And then based on the data we got from that, we did further rounds of selection really of the vaccine candidates to identify the conditions that generated the most stable vaccines. So The second round looked at 6 iterative formulations based on what we learned from the first round, and then we did also a final round of selection, and finally demonstrated reproducibility for the lead formulation for each vaccine candidate, and demonstrated vaccine vial monitor 14-day stability at various temperatures. So just again, the 2 vaccine candidates that we focused on: receptor binding domain of the spike protein of SARS-CoV-2, and a malaria vaccine target displayed on the surface of a virus-like particle. So we took several different assays to look at the vaccine candidates. So we established assays to analyze both solid and solution state characteristics of these spray-dried vaccine candidates. So both looking at the dry powder characteristics, such as appearance, moisture content, particle size, surface morphology, and then also the reconstituted in the liquid state, looking at the integrity, identity, concentration, purity, aggregation state, and activity of these vaccine candidates. For the first round, we looked at 24 formulations for each vaccine candidate, 12 with, 12 without an adjuvant present in each case, and looking at varying the sugar contents, other excipients, and also the buffers used, and the temperature of conducting the spray drying. And we went through 3 rounds of selection of vaccine candidates here, looking for the preferred formulations. And what we got out of it was that we managed to develop a spray-dried formulation for the protein subunit vaccine, the receptor binding domain, with vaccine vial monitor day 14 stability at 60 degrees C. And then for the virus-like particle, through a similar process, we developed spray-dried formulation of the VLP with vaccine vial monitor day 14 stability at 37 degrees C. Out at 60 degrees C, we did see some disintegration of the VLP structure, but it was stable for 2 weeks at 37 degrees C. And we also showed reproducibility for the formulations, for the lead formulation for both of these vaccine candidates. So the key findings from this project, first of all, we found that some assays that we developed were incompatible with the inclusion of specific excipients or adjuvants, requiring the development of alternative assays or the down selection of particular formulation components. With regard to identifying the preferred excipients and spray drying conditions, we found that trehalose and gelatin were the preferred excipients in our case for improving the dry powder characteristics of these vaccine candidates, and also the stability of elevated storage temperatures for both of them. However, a high sugar content was detrimental to the stability of the virus-like particle at elevated storage temperatures. And then with regard to the spray drying temperatures, we found that in general, higher temperatures positively impacted the solid state characteristics for both vaccine candidates. But temperatures of 190 or even to some extent 150 did lead to some degree of virus-like particle disassembly. And a temperature of 190 led to some loss of activity of the receptor binding domain when it was subsequently stored at an elevated temperature of 60 degrees. So out of this, we gleaned that the best temperatures to do this for us were around 110 for the virus-like particle and around 150 for the receptor binding domain. So the key point to take home from this is that we did develop spray-dried formulations for the receptor binding domain and the virus-like particle vaccine candidates with vaccine vial monitors day 14 stability at 60 degrees and 37 degrees C respectively. And then looking forward for possible paths to take this on these candidates or extend to other vaccine candidates. One obvious thing would just be to extend the stability testing for these lead spray-dried formulations out to vaccine vial monitor day 30 at the same temperatures that we identified as being acceptable for the receptor binding domain and for the VLP. Most importantly, to assess the immunogenicity and protective efficacy of these lead spray-dried formulations in appropriate animal models to ensure that they perform at least comparably to the liquid formulations and that therefore they will be suitable for further development. Potentially to extend formulation screening to include additional bulking agents that for just reasons of cost and time were not included under this project. And then from the point of view of vaccine design, looking at doing a spray-dried formulation of a virus-like particle, the general design looked at under this project, but instead of displaying a malaria vaccine antigen to display the receptor binding domain of the spike protein of SARS-CoV-2 or other coronavirus antigens on its surface. So this would be a VLP targeting COVID-19 and would bring together the two different aspects of the targets looked under this program. And then more generally, just to extend the types of spray-dried formulations developed under this project to other protein subunit and VLP vaccine candidates to assess the general applicability of the spray drying approach for vaccine formulation and development. And that wraps up the presentation.
Thank you, Steve. We do appreciate the presentation, and we do have a couple of questions we wanted to ask you. So what are the business benefits of providing more thermostable vaccines? For example, does it simplify the supply chain, reduce product destruction events, anything like that? Can you describe those business benefits?
I think from a business standpoint, it would be reduced cost to run the spray drying compared to lyophilization. Certainly reduce time and energy requirements there. And it can and then from the point of view of the vaccines that you generate, there's that reduced cost aspect. And I think there's a lot of work now to try and scale these technologies. And the fact that they can run in line with other downstream processes just makes it more amenable to longer term, I think, to being incorporated into downstream processing.
Excellent. Were there any new methods and tools of analysis generated as an outcome of this project?
No, frankly, we use fairly standard biophysical tools, looking at dry powder characteristics, and then also the liquid formulations when the reconstituted formulations, also just looking at fairly standard biochemical assays. And we did that because we had, there's a standard array of assays that are typically used for the lyophilized products anyway. And then we had well-established assays for these particular molecules in the liquid state. More generally, the project was more about just identifying preferred conditions and components for spray drying of these particular vaccine candidates, particularly extending it from protein subunit vaccines to more complicated VLPs.
Great. And lastly, are there any publications on the horizon documenting the success of the project?
Yeah, we're looking at doing that both for the spray drying aspect of specifically what was done under this project, of the more stable formulations that we developed, and then also what I talked about at the end of some of the sort of paths forward, looking at a VLP combining aspects of the of displaying a COVID-19 antigen on the surface of the VLP, and then looking at how that performs in comparison to just the protein subunit vaccine.
Great. Thank you, Steve, for the presentation, and thank you for watching our American Rescue Plan webinar series. To find additional webinars, visit www.nimble.org.